Okay, we're going to quickly move into our panel discussion, our research panel Q&A. I need Jeffrey Holt, Jen Lentz, Dr. David Gamm, Alex Levin, and John Sholar to all come up here. We're going to get you some chairs and microphone for you.
I brought my own.
You got your own. You're awesome. Okay. And so we gotta try to wrap it up at 12:25.
I like the proactiveness of everybody.
Yeah, we all brought your own chair.
Okay, good morning again. I'm Jeff Holt. You saw me earlier. I'm a scientist at Boston Children's and Harvard Medical School. I'm up here now with our beautiful panel of speakers who joined us already. I know this morning we've thrown a lot of science at you, some complicated science. Our goal has been to explain the state of the field in terms that would be accessible to a broad audience. But there may be questions, and actually this is the time to ask them. So we'd love to hear from the audience. We've got speakers here. If you have a question for any one of our speakers, you can address them specifically. If you have a general question, we'll try to do our best. I'll select somebody here on the panel to try to field that question. And if you do have questions, please raise your hand. We have microphone runners who will deliver microphones so that you can ask your question. Please identify yourself. If you want to share an Usher diagnosis or an Usher form that you want to hear about in particular, please feel free to do so. So with that, I'll open to questions. I see one over here.
Looks like
to
we
the
have
left.
one right here.
Oh, okay, we've got a microphone already in the back left corner. Please go ahead.
It's not on.
Hello, everyone. My name is Arthur. And I am from Ohio. I wanted to explain a little bit about my experience from 1987. I went to an RP workshop in New Jersey, and while I was there were several panelists. Several of them were researchers, and they were hearing and they said that there were 33 different forms of RP. And so now I'm wondering if that is still accurate. Are there still only 33 types, or have new types, new forms been added? Or maybe there are fewer than 33 at this point. I guess that's my question. Was my question clear to all of you?
Yes, thank you for the question, and I think Dr. Levine might be in a good situation to address that one.
1987, there was actually only one form of RP. That was the rhodopsin gene, which was the first gene that caused RP, wasn't discovered until 1990. Now, there's well over 100 different forms of RP, if not over 200 different genes which are involved in RP or RP-like diseases. And still more genes are being discovered.
Great, thank you very much. We have
So how many known forms are there now?
Over 100 known forms right now.
great, thank you.
Wow.
I see another
Thank you so
question
much.
here in the front.
Hi, this is Alan from Those Meetings.
Thank you. This is for Dr. Levin or Levine?
Anything works.
Dr. L. You had mentioned, obviously, it's very important for people to be cautious and to be skeptical and make sure that they do their research. But you specifically said something about optogenetics, and I know that there are active human trials going on right now, and how that might be particularly dangerous or potentially dangerous. Can you elaborate on that a little bit? I hadn't heard too much about
So
that.
first of all, we don't know yet what vision optogenetics can give us. It's probably going to be poor vision. So the candidates for that are not people who are 20/80 and 20/60, and that's going to be for patients who are really, really poor vision, like no light perception or light perception or just seeing a hand moving. The early trials, there's one trial from India which was encouraging. It is an injection of a gene into the retina. So there's a physical trauma of that, and it's way too early to know whether it's worth that trauma. If you take, for example, the FDA-approved treatment of an electronic chip on the retina to restore vision to people with no vision, that chip is great. It gives you light perception, but if you go to the people who have that chip, they only turn it on about 20% of the time. That gives you an idea of the efficacy. And whether optogenetics is going to be in that realm or just give some navigating vision and it's worth it to inject your retina, which will likely preclude the opportunity to get a gene therapy trial that's more specific or directed later on, is yet to be seen.
Great, thank you. Another question over here for our microphone runners. Right up front.
Should we just hand this one over? I can run.
All right, it's working? Excellent. So I'm curious about research into comorbid conditions with Usher and how that might affect treatment and clinical trials. My name is Elliot. I'm from Canada. I was only diagnosed recently, and I've also been diagnosed with autism since I was very young. And so I'm curious about research into Usher syndrome and autism, Usher and ADHD, and other, like I said, comorbid conditions and research into that.
Great question. Anybody want to, Marley, you want to take a stab at that?
I mean, I think a lot of us can probably answer you on that. So there are inclusion and exclusion criteria. We have a clinical trial coming up in 2025 as well. But those would not preclude you from being involved. So the sorts of things systemically that we're interested in would be things that would make it more dangerous for you to undergo the procedure itself, right? Or that might possibly confound something that we're looking for. So, and those are important. So you might think, oh, I don't want to be excluded, but usually if there's a group that's being excluded early on, it's so that we get the cleanest signal we possibly can, so we don't fool ourselves or anybody else. So there's a reason for it. But I'll tell you that a lot of at least the stem cell-based trials thus far have been for macular degeneration, which have been in patients who have been very old, so 70, 80, 90 years old. So there's a lot of things that go on when you get to be 70 or 80 and even 55. So, clearly there's some buffer there that allows you to be able to participate. But then some folks have an MI and no one wants to put anyone to sleep for that. So it also matters what type of therapy you have. If it's bedside, if it's just an injection, it's just like J-site, then they may have less of an interest in what your comorbidities might be, as opposed to something that required anesthesia.
I think first of all, I'm Canadian as well, so welcome. Glad to have you. I think your question may have also been asked about research into the association of these conditions. And there's two ways to answer the question. First of all, common things are common. Usher syndrome is rare, but just because a person has Usher syndrome doesn't make you immune to all the other things that are more common in the world, from the common cold to autism to ADHD, et cetera. So when we look at comorbidities in people, we're always wondering, are they just coincidence because you have a common thing? Or could it be really related on a genetic basis? For the genes that have been shown here in USH1, USH2, USH3, we're not aware of any specific relationship to autism or ADHD. But there are other forms of hearing loss with RP that can have other comorbidities with them. And that would be revealed on further genetic testing. If you already have a genetic test and you're one of these diagnoses, then it's unlikely that that's related.
Okay, great. Marley, did you want to
Yeah,
add?
no, those are great points. I was just going to say the other thing is for any type, not just gene therapy, but for any type of therapy that might be for the vision impairment, or since I'm an ENT person, the hearing loss, you want to make sure whatever potential side effects of that therapy they might be looking for aren't things you already have. So if, for example, you had some sort of irritable bowel thing and the side effect is nausea and vomiting, it would be impossible to sort out. So this would be one of the things they would hopefully ask when people enter any type of clinical trial for anything, whether it's local therapy or systemic therapy.
Yeah, I think one other point on that, just so I want everyone to be hopeful, is whether or not someone is in the inclusion-exclusion criteria for the trial usually doesn't have a lot to do with whether or not the drug might work in that population, right? So, just because you can't get into a trial, don't lose hope. The label will be whatever the label is from the agency down the road, and there's a lot of conversations that come with those thoughts.
One last comment along the lines of research and a better understanding of comorbidities. Carmen Trupig is going to talk this afternoon about a large push to conduct and collect data from individuals with rare diseases, including Usher syndrome, about all the symptoms you're experiencing. And this is because we want to know what else individuals living with Usher syndrome may be experiencing. So listen up for Carmen later. And also in the back of the room, you can talk with Nancy and Monica about how to sign up for and take those surveys and participate, and let us know about your symptoms as well.
Great, thank you. I see a question over here on the right.
Speak.
Okay. Hi there. My name is Lisa Flaherty Vaughn. I am deaf and low vision from Connecticut. Well, I actually was born and raised in Massachusetts. And so I go into Boston for diagnostic testing and everything. I've done that since I was 7 years old. So as a first-time participant, I've felt a little bit lost because I hear people talking about Usher syndrome. Well, first of all, I want to thank you all for being here. Thank you for your research. And thank you for your interest in helping resolving a lot of the issues that and making our lives better for people who experience Usher syndrome. I learned that there are varying types of Ushers. There's Ushers for that people who don't have any hearing loss, and then there's mild hearing loss, and so on. So, let me say, I was born deaf and blind, obviously, and I have Ushers, and it is genetic. Is it possible that some of the treatments or experimental treatments you mentioned would work on a person who was born deaf and blind?
Thank you for the question. Anyone on our panel would like to field this one?
I'll take the hearing.
You do the ears, I'll do the vision.
So that's a wonderful question. And I think it has a lot to do with the therapies that will be utilized and what the target cells are, and I'm talking about the ear. I think for people who have been deaf a long time or have cochlear implants and their implant is working for them, people ask if the hair cells are still like in there and possibly being able to be stimulated. I think the short answer is maybe. It would depend hugely on what the therapy was, though, and whether there was the possibility to either grow or stimulate the hair cells that are already there, as opposed to bypassing them, which is what cochlear implants do. I think the eye question might be different, though.
It's actually very similar. So if you take Luxturna, the treatment for a cause of congenital blindness, those kids really do see again. But in that disease, you're born with the cells. They're all alive, they're just not working. Usher syndrome, you rarely are born blind, but you can have early vision loss, cells are dying. And you would have to get in there early to keep those cells alive. The rest of the retina, though, remains healthy. And if we could figure out ways to put stem cells in, to take advantage of that remaining healthy retina, then conceivably you could restore vision. So I think the possibility is certainly on the table, and we have an example already from one disease where vision can be restored.
Great, thank you. Other questions from the audience? I see some back here to the left side of the room. Can one of our runners head over? Yes. Sure. See. Microphones are on the way.
All right. Hi, everyone. I'm Nicole Alleman. So it's good to see you again, Jennifer. Thank you for the panel. This is wonderful. I need you all to identify who you are when you're responding to questions, because I don't know who's speaking. So please, if you could, identify yourself with your name before you respond to a question. We would all like to know who's speaking. Please. Thank you. So keep doing the wonderful work that you do, and please identify yourselves.
Great, thank you for that. That's a good idea. I'm sure our panelists can help with that. Let's go to the back here.
Hi, thank you for being here. And my question is, I'm a parent of a child with Usher 2A. When I brought her to a doctor, I'm lucky, I'm in Boston, I immediately had the best doctors. And when we first brought our daughter to a doctor, the vision retinal specialist, we were told in about, a decade there would be a treatment, at least to stop the progression. Now my daughter's almost 13, and she has a good amount of night blindness, and there is no real treatment. So I think my question is not so much to say, hey, what happened? Because I get it, it's slow, it's complicated. Couldn't even begin to understand how complicated it is. But when I have my child and I listen to all these great things and she's not eligible to do anything, what is it that and I know it's slow progressing, but what is it with the people that are kind of caught in this situation, maybe the parents with the younger children? Is it NAC attack that we should be looking at? What is it that we can do to slow the progression so that by the time the treatment is available, whether it be J-site, which seems to also slow the progression, or ultimately what we all hope is some sort of genetic treatment to completely stop it. What is it we should be looking at and doing? Because I feel like I come here every time and I'm very hopeful, but I also feel like none of it's for me, or none of it's for my daughter yet. But I'm now watching my daughter lose her vision. So I guess that's a hard question, but where should we be looking at? Should we be looking at anything, or are we just waiting?
Hey, Alex, do you want to take that one?
Sure. This is Dr. Levin, Alex Levin. So it's hard when you have a child and you see things moving fast, and you're often not eligible because you're a child, right? Many of these studies have cutoff points at 18 years of age, sometimes a little younger. There are a few things that we know are beneficial for the retina in general that I tell all my patients with RP. Don't smoke. Obviously, it's a child, but if your parents smoke and you're around secondhand smoke, that's bad for your retina. Number 2, eat a well-balanced diet. Number 3, fish is good. DHA, which is a chemical in fish, there's some evidence that shows that can slow progression. I recommend patients who have RP eat fish 2 to 3 times a week, preferably fatty fish. If you like sushi, it's a good excuse to eat sushi. And if you don't eat fish, you can take DHA, is available in algae tablets. Wear sunglasses outside. The ultraviolet rays of the sun are damaging to your retina, and you don't need extra damage to your retina. Is it something that I would battle with my child over? I don't think you have enough things to fight about with your child. But those are kinds of things that we know are good retinal health. As far as what trial to get into, it gets back to my talk. You have to see if you're eligible and whether you want to go through all the stuff that's involved in doing a trial. I would personally say that if I had a young child, research is happening quick enough that I would be slower to get involved in a trial. Let the adults do the trials. And I think that by the time things come around, there's hope that there'll be something for you.
David Gamm, just to reiterate that too. So when I tell my patients, I hear that an awful lot, right? But there are risks involved. And so as a parent, when you don't know what those risks are, would you want to put your child in a situation where they may lose vision and it's your decision too? So it's a heavy thing to do. And so whereas when they're 18 or above, they can make their own decisions, they can consent as opposed to assent. So I often say for something that we don't know the safety profile for yet, being second in line, third in line, watching to see what happens to folks who are oftentimes maybe too far along in their disease to really benefit tremendously from an efficacy standpoint, but at least we'll be able to see whether the safety profile is good enough for us then to put our children potentially in harm's way. So following that research, everything that's being done in adults will filter down to children eventually, and I think appropriately later on, because I think there's more to lose with kids as well too.
Yeah, this is John. From J-Site, from an industry perspective, we always want to start with the adults if it makes sense. And of course, it's part of the plan every time to quickly, expand the label and do additional trials in a pediatric population after the fact. But, we want to land it first and make sure we've actually proven the therapy works, and then we think about moving it along. Now, which isn't to say we can't move it along quickly. I mean, the agency in rare disease is being very, very cooperative on that point. But I think our view is get the adult indication, done and dusted, and then talk about what we're going to do with pediatrics.
Wonderful, thank you. Other questions from the audience? I see one way back here in the right, if we can get a microphone runner.
Hi, my name's Abby. Ooh. I'm diagnosed with USH2, but I was just wondering, is the vitamin A kind of a, if you prefer? Because I know Susie was talking about it and said that there was no, it doesn't benefit it. But I was just looking at the website, and it says if you've been taking it for a while and you're satisfied with it, keep taking it. I've been taking it for about 10 years, and I asked my eye doctor, and he didn't want to tell me no, which I understand, because if I stop and something happens, that falls back on him. I get that, so I'm just wondering if the vitamin A, is that just a preference, not really recommended? If you just want to take it, you take it? And thank you for your time.
This is Alex Levin. So I would venture to say that 95% of people who specialize in RP are no longer recommending vitamin A. The trial that was done, unfortunately, there was a lot of things that were wrong with that trial that showed up over time. Vitamin A can be toxic to an unborn baby, so women of childbearing age have to be aware of that. There's also some systemic risks to the rest of your body that need to be considered. And the original studies weren't done with respect to specific genes, and therefore we learned that there are some genes in which vitamin A makes you worse. So we don't, at least in my practice in our group, we don't recommend vitamin A for patients with Usher. If you're taking it and you're doing well, that doesn't mean that it's safe, and you need to be monitored by your medical physician for the side effects of this disease with regular blood work, and to be practicing birth control so that it's safe for an unborn fetus.
Okay, thank you. Others, I see some hands over here to the left. Do we have our microphone runner on this side? Here she comes. Microphone's on the way.
Hi there, my name is Ronald Boucher. I grew up in Maine, and I'd like to applaud all of you for the research that you do. It's very interesting and fascinating to me. Now, for Jennifer, would you mind sharing your PowerPoint with me or with all of us so I can share that information with my parents?
Yes, I'm happy to do that. I think this is Jennifer speaking, Jennifer Lentz. I think our presentations will be made available on the Usher Coalition website, but we can also provide PDF versions, and I'd be happy to email you.
Okay, thank you.
Great, yes. Another question back here to the left.
We're gonna, yeah.
Hi, I'm Mark Wallace from Chicago, Illinois, and thank you so much for everything you do. It's really inspirational to be here today. So just a quick question. One of the things that I heard a few times that kind of pricked my ears that I'd be interested in your views on is this is clearly moving very quickly thanks to the good work that you're doing. And I've also heard some feedback, well, you should be careful about considering this because it might preclude participation in the future or those types of things. So I'd appreciate any feedback from this group in terms of if you're evaluating whether or not to participate in one of these trials, some pointers that would avoid any issues in the future or any conditions to consider in terms of, would I be a good candidate or not? So thank you.
So I'm happy to start because I've got a trial starting
Go ahead.
in a little while. I think the way we look at it is this. It really depends on the therapy involved in the trial that was sort of the first trial, whether or not you'd be eligible for a second trial. I don't think anybody thinks taking NAC is going to be a problem in another trial down the road. Speaking specifically about J-cell, we know it frankly disappears after about 18 months, and it's an intravitreal injection. We're not touching the retina. So our view is, you can be in another trial after ours and there's no problem. That said, that's not my decision to make, right? So, I can tell you what I think about what will happen in a subsequent trial with a different gene therapy or something along those lines, but, they're going to come up with their own requirements down the road. So while, my chief medical officer says this won't be a problem down the road, somebody else's might have a different point of view. So there is an inherent risk there, but we're pretty comfortable.
This is Alex Levin. I think that almost every single trial, if you've had a previous intraocular injection or surgery, will eliminate you from a secondary trial. That's during the clinical trial stage. It may not eliminate you when it becomes a treatment and it's approved, but it will almost certainly eliminate you from another trial. They're all worded. I mean, we all do them. We construct them. We make our inclusion criteria. It's just because you don't want to. If something happens wrong, is it due to the first thing? Is it due to the second thing? Is it immunological response and you get inflammation in your eye? Is it due to number one, number two, or the combination? So most of the time, the one surgery that is proving to probably not get people out of trials is cataract surgery. Most studies will allow you to still go in if you've had cataract surgery. That's about the only one that you can get into a trial with having a previous eye surgery.
Yeah, this is David Gamm too. So I agree with Dr. Levin. I don't see anybody receiving a cell-based therapy to the eye that would be eligible for another phase 1, phase 2A cell-based therapy. There's just too much potential overlap. It's an exclusion for ours as well. And other things too, if you think about it, if you've got a history of retinal detachment, you're going to be excluded from that too, because if you're putting something under the retina, at least that potentially could detach the retina. So was it because of the surgery, or is it because you had a predisposition to it anyway? So you do have kind of in many response. It's a deep question, because you may have one eye to give, right, for a clinical trial. So you have to kind of think that through. But then you might also be waiting and missing a window where a particular therapy might be useful for you. You're waiting for something to come by that's going to be bigger and better. And as you've seen, it's all kind of shades of gray, right? So you have to, talk to your doctors and talk to your family and decide what's best for you. And it's not going to be black and white, unfortunately. We wish it would. But I do also think that in the end, if it's approved and it shows efficacy, then it'll be open to folks whether or not they've had previous trials.
I know the
Marley?
The question is probably for, I'm Marley Kenna, sorry. I talk a lot, I should be able to tell you that. So I think the question was mainly about the retina, but from the hearing loss side, since the hearing loss is often what's diagnosed first, we get a lot of questions about folks, especially with USH1, should they do cochlear implants or should they wait? And we're very fortunate that we have cochlear implants, as I think we have witnessed here, and many patients are extremely successful. So I think, but there's also a window, if you wait until the patient is 18 and the patient is born deaf, we've lost the window of opportunity to do anything, whether it's gene therapy or NAC or cochlear implants. So it's a decision we have to make at a much younger age. Having said that, it would almost really have to be as good or better than a cochlear implant. And so we're actually doing a gene therapy trial now at Children's. It's not for an Usher gene, but that's our criteria.
Great, thank you. There's a question over here on the right. Can we get a microphone up front?
Hi. Wow.
Okay.
I'm a grandmother of a 13-month-old with USH2. And my question is, as a community, first of all, we appreciate everything that you're doing, and I know that it's a slow process, but as a community, are there things that we can do to help you to speed things along, whether it's reaching out to the rare disease governmental agencies or anything like that? What can we do as a community to help you?
Great question. Who would like to field that one?
Happy to take David Gamm again. So really, a lot of it more and more is related to your representatives and your home states. So what we're seeing is that the National Institutes of Health, which don't fund a lot of the big trials, they go to industry, but the research that's done that spurs almost everything that we see starts in a laboratory somewhere in most cases. So that's, or is advanced in increments in laboratories. And that funding comes through government agencies, foundations like this one, which is really important to get us going in the right direction. But more and more we're seeing politicians taking that over and telling us, you can only work on this or you can only work on that. Well, they're choosing those things based upon who's whispering in their ear. So if nobody's whispering in their ear, hey, Usher syndrome's important, and it's scary, right? Because that's how you get things diverted to pet projects. That's how you get some projects that make no sense whatsoever, but some senator somewhere gets it in their head that it's a good idea. And by the way, they also didn't pass 10th grade biology. And so they're the ones telling everybody what to do, and in that regard, telling you what you have available for you. So interacting with your local and state politicians is important now, and will be more so going forward as Congress seems to think that they know more than the folks at the NIH do.
I think you're helping by being here right now. Just by creating a community and working together to create awareness, to get people interested in the field is really important. Fundraising is critical. It really is. I mean, there is not unlimited funds for research. As I said, our mission is entirely philanthropically driven. That's important, and politics is important. So I think you should all congratulate yourselves on getting out of bed on a Saturday and coming here, because you're moving the agenda by being here.
I think also you can't underestimate that you think you're one voice and you can't make any difference. I think that's so not true. Parents, on behalf of their children or people who are living with this challenging situation, you have to be loud and maybe a little bit obnoxious, but you can make an enormous difference. And for people who think they can't, we're here to say, obviously, you absolutely can do it, and I applaud all of you, and even on a Saturday.
Yeah, this is John. So I think one of the interesting things about the panel is this is a really long race where you hand the baton off from the basic science to the translational work to the industry trying to get a drug approval. And so everyone's perspective comes at it a little different. When I think about drug approval, I think about what happened with Duchenne's, 12 months ago now, right? And that's all about patient advocacy at the agency, and those patients going in and saying, and their parents really in that instance saying, we want this therapy approved. And frankly, I think Peter Marks just said, if you want it, you can have it, right? And we can argue about the science behind that and whether that's a good or a bad thing, but it very clearly made a difference, I think, in the Spark trial, and I think it very clearly made a difference for Sarepta and Duchenne's to have patients there when you're going, patients and their advocates there when you're going to those FDA approvals. So, I think it's just as important at the beginning, but at the end, you see a pretty direct impact when maybe something's, call it a coin flip about whether the agency's going to approve it or not.
A great place to get ideas, by the way. There's a book by Ricky Lewis. It's called The Forever Fix. It's about really parent advocacy and how it's made a difference in getting progress in a variety of different diseases. Highly recommend it.
This is Jennifer Lentz, a researcher. I would also encourage you as parents and individuals living with Usher to consider participating in data collection. Your experience with your Usher is just as important as the research we're doing in the laboratory to develop new drugs and get them approved. So reach out to those that are conducting natural history studies or the data collection program and consider if it's the right thing for you.
Okay, great, thank you. I'm seeing that Krista is signaling to me that we have to stop the Q&A session. I'm sorry if you did not get your question answered. I would invite you to find some of our panelists during lunch. They'll be around. I'm sure they'd be happy to speak with you all. And I would like to thank all of the panelists for being here and for their wonderful and insightful questions. Thank you very much.
We're just going to have you stay here for one moment, reminding parents to pick up their littles, the 0 to 8s, for lunch. And we need to be back here at 1:55. I'm going to have Dr. Kumar come up now.
So good afternoon, everyone. We have come as a team for this Usher Coalition Group meeting from India, despite Microsoft. So it's almost about 11 to 12,000 kilometers we have traveled, and we have Mr. Agachand here, and then we also have Mr. Ramesh, his brother, and also Mr. Panna. And the purpose of our visit is to make Mr. Agachand know what is going on in research in Usher syndrome. Okay, so we have come all the way with Mr. Agachand, who has retinitis pigmentosa, and he also has DNA variants in USH2A gene. And he wanted to know what's going on currently. And the second purpose is that we want to take this information to our patients in India with Usher syndrome. And all of you know that we have about 1.4 billion people, almost 3 to 4 times what US has. So many patients there can be benefited through this coalition group, and we can do similar activities in India. The third purpose is that there are many scientists who are fighting blindness across the world, and we are also a group in India who are doing this by doing research. And we are setting up a gene therapy lab in our hospital in Bangalore, and the real purpose is to take this to India. And again, as Dr. Levin said, the cost is a major factor, and therefore we would like to collaborate with him so that this is easily available in India for people who can afford as well as people who cannot afford. So because of the research and clinical services that Dr. Levin has done through his lifetime, we wanted to honor him, and particularly Mr. Agachand wanted to honor him. So in India, what we do is that we have a traditional practice of putting what is called as a shawl. I'll explain what this is. It has been for centuries that we honor people who have done good service to the community. And this is a cloth material which in the olden times was actually laced with gold. And because gold has become very expensive, today we have silk cloth which we actually try to put it on the person whom we wanted to honor. In fact, we wanted to honor all the people sitting on the dais who have done considerable amount of research to help people with Usher syndrome. But we didn't plan for this. We have only one shawl, and we'd like to put it to Dr. Levin, Professor Levin, because of his lifetime service that he has done. And that will be done, the honor will be done by Mr. Agachand.
Right, great. We'll see you back here at 1:55 after lunch. Thank you.